to predict toxicity, corrosivity, and other safety variables as well as the effectiveness of a new product for humans. Traditional testing of chemicals, consumer products, medical devices, and new drugs has involved the use of animals. But today, scientists have developed and validated alternative methods shown to lead to safer and more effective products and drugs for humans than animal testing. For example, skin corrosivity and irritation can be easily measured using three-dimensional human skin equivalent systems such as Epiderm and Skinethic. Additional alternatives include Episkin, a model of reconstructed human epithelium, and a variety of sophisticated, computer-based quantitative structure-activity relationship (QSAR) models that predict skin corrosivity and irritation by means of correlating a new drug or chemical with its likely activity, properties, and effects with classification accuracy between 90 and 95%. 1. Some alternatives to the use of animals in testing include in vitro, test tube, test methods and models based on human cell and tissue cultures, computerized patient drug databases and virtual drug trials, computer models and simulations, stem cell and genetic testing methods, non-invasive imaging techniques such as MRIs and CT scans, microdosing, in which humans are given very low quantities of a drug to test the effects on the body on the cellular level, without affecting the whole body system. Benefits of non-animal testing. Alternative scientific tests are often more reliable than animal tests. For example, experiments on rats, hamsters, guinea pigs, mice, monkeys, and baboons revealed no link between glass fibers and cancer. Only after human studies related the two did the Occupational Safety and Health Administration (OSHA) label these fibers as carcinogenic. Epiderm, an in vitro test derived from cultured human skin cells, was found to be more accurate in identifying chemical skin irritants than traditional animal tests. In comparison studies, Epiderm correctly detected all of the test chemicals that irritate human skin, while tests on rabbits misclassified 10 out of 25 test chemicals, a full 40% error rate. The use of human tissue in toxicity testing is more accurate than the animal models. The lethal dose 50 (LD50) test forces animals to ingest toxic and lethal substances to the endpoint of where 50% of the animals in the study die, and those that do not are later killed. The late drive. JRN Ekwall, Cytotoxicology Laboratory in Sweden, developed a replacement for the LD50 test that measured toxicity at a precision rate of up to 85% accuracy compared to the LD50 rate of 61 to 65%. This test, far more accurate than the animal models, uses donated human tissue rather than animal. Further, the test can target toxic effects on specific human organs, whether or not the toxic substance permeates the blood barrier, and other highly sophisticated and precise information that the agonizing death of an animal of a different species would not reveal. Non-animal tests are more cost-effective, practical, and expedient. In vitro international sclerocytics, synthetic skin, can provide a chemical corrosivity determination in as little as 3 minutes to 4 hours, unlike animal testing that often takes 2 to 4 weeks. The ACDEC, an alternative test used to measure the effectiveness of sunscreens, was reported to do in days what it takes animal studies months to do, and estimates that it can test 5 or 6 products for less than half the cost to study a single product in animals. The traditional testing of chemicals using animals can take to 5 years per substance and cost millions of dollars, while non-animal alternatives can test hundreds of chemicals in a week for a fraction of the cost. Cruelty-free products are more environmentally friendly. In toxicity testing, researchers breed, test, and ultimately dispose of millions of animals as pathogenic or hazardous waste. Cruelty-free testing is less harmful to the environment or creates less waste. In 2007, the U.S. National Research Council (NRC) released a report titled Toxicity Testing in the 21st Century, A Vision and a Strategy, which addressed the limitations of animal-based toxicology tests and called for a shift toward non-animal, human-based testing methods. The report was issued in response to a request from the U.S. Environmental Protection Agency (EPA), asking for the NRC to conduct a comprehensive review of toxicity testing methods. In the report, the NRC summarized that the advances in toxicogenomics, bioinformatics, systems biology, epigenetics, and computational toxicology could transform a system based on whole animal testing to one founded primarily on in vitro methods that evaluate changes in biologic processes using cells, cell lines, or cellular components, preferable of human origin. 2. In 2008, the EPA and the NIH's National Toxicology Program and Chemical Genomics Center signed a Memorandum of Understanding. to follow the NRC report's vision and begin developing new methods of toxicity testing that involve the use of lab-grown human cells instead of animals. After the EPA began evaluating 300 chemicals using the new methods, they found that it allows for thousands of chemicals to be tested at once. This method is much faster, less expensive, and does a better job of protecting human health. According to former NIH Director Elias Zerhouni, it won't mean that animal testing will disappear overnight, but it signals the beginning of the end. Today, the EPA is in the process of building virtual human organs. The EPA's virtual tissue researchers are developing a set of computer simulations that may one day be able to identify the risks posed by common industrial pollutants such as pesticides, saving thousands of animals from horrific toxicity tests. 3. ICCVAM. The need for alternatives to the traditional use of animals in toxicity testing was officially recognized by the U.S. government in 1993 with passage of the NIH